Okay, our first unit is called Resist, the Biology of Antibiotic Resistance, and part 1 hopefully starts to answer the question that many of you had, which is, how does this happen? Because it's horrifying. And it seems like most of you had not really heard much about this prior to a couple days ago, though I would bet that some of you know somebody who had MRSA. How does this happen? How does this horrible thing actually happen? That's what we're going to start to answer. Okay, here's the format I'm going to take. I'll read the slide, then I'll shut up for a few minutes so you can finish copying. Background. Antibiotics are a specific type of drug that kills bacteria. Some bacteria are not killed by antibiotics. Bacteria that aren't killed by antibiotics are called resistant. Resistant bacteria may be resistant due to slight differences in their cell structure. Bacteria's cell structure is determined by the genes contained in their DNA. Now, the other point that I want to make, the words on the slide are really only a fraction of what you need. And as we're talking about sort of note-taking, it looks like probably over half of you have plans to go to college somewhere someday, doing something. And when you get to college, there will be a lot of notes. And a lot of it is just walking in with a prof who draws a couple things on the board and starts talking at you. And you figure out what's important and write it down. If you don't drown first. So I give you some bullet points, and these are as much my jumping off points as they are important pieces for you. When we're talking or I draw something to illustrate an example, you should include that in your notes. That extra information is meant to be helpful to you. Okay? So, antibiotics are a specific type of drug that kills bacteria. If we think back to that whole root and prefix and suffix, what does anti mean? What does anti mean? Against. What does bio mean? Life. Guess what antibiotics are? Drugs that are literally against life. They kill living things. Whoa, do I want to take that? Now, these are drugs that kill living things, which are little tiny one-celled organisms. We're going to talk more in a little bit about what bacteria are. Can an antibiotic damage your cells? Yes. Yes. What was the drug that we heard about in the movie that damaged David's cells? If you don't have to remember it, it's fine. I'll just be really impressed. Colistin! Yes, some of you got it. It's an old antibiotic, and it sure does kill cells. It kills bacterial cells. It was also destroying cells in his organs. It was attacking his heart and his intestines, and it was really making his life hard. So it is anti-life. It is against life, but you have to have a drug that will target the kind of cells you want to kill. You don't want a drug that will target all living things. Has anybody ever disinfected something with chlorine bleach? Anybody got a pool or anything at home, or they've been around a pool? I grew up with a mother who was more of a, I'm probably an anti-germaphobe in response to my mom, because growing up, the entire bathroom always smelled like Lysol. She likes to Clorox public toilet seats. I kid you not. She's a little freakish about this stuff, and I'm like, it's fine! Chlorine is potently anti-life. It'll kill all kinds of cells. It'll kill bacteria. It'll kill viruses. It'll kill, well, we'll talk about what viruses are. They're not living things. It'll kill protozoans. It'll kill amoeba. It'll kill an earthworm. It'll kill cells in your hands. If you're exposed to strong solutions of chlorine for long enough, you'll end up with little dead spots on your fingers, little skin cells that have died. Potently anti-life. However, would we want to administer chlorine intravenously to cure a blood infection? No, I don't think so. That would tend to be fatal for the patient. So antibiotics are a class of chemicals that are drugs, which means we can put them into a human body, and they'll attack the bacteria, and hopefully, outside of things like colistin, they won't destroy the person. There are some bacteria that aren't responsive to antibiotics. And we saw that in the film. Has anybody here, well, no, I won't ask that because it could be embarrassing, but I am going to guess that at least somebody in this room has had MRSA or known someone who had MRSA. Like I said, we had the year when everybody who used the weight room seemed to get MRSA. MRSA is a bacteria that's not killed by common antibiotics. It's methicillin-resistant Staphylococcus aureus. Whoa, it's a mouthful. MRSA's easier. When there's a bacteria that isn't killed by an antibiotic, we call that a resistant bacteria. And resistance is typically not all or nothing. So resistance can be to one antibiotic. I'm going to abbreviate that by anti-B, but not another. And if you remember, they had that little chart when they were looking at David's results, and they said, you know, all these R's mean resistant, all these S's mean sensitive. So you can have a bacteria that's resistant to this antibiotic but is killed by this antibiotic. What we worry about is when we see drugs that are, what's that term for resistant to all antibiotics? Pan-resistant, yeah. And we had Addie's mom saying, you know, they told me pan-resistant. That meant everything. Like a panorama is a picture of everything. Pan-resistance means we can't find an antibiotic that this thing responds to. How can an antibiotic resist a drug? Or how can a bacteria resist a drug? How can it resist? This is terrifying. What did he say about the gram-negatives? Do you remember? That's just a particular class of antibiotics, that, or a class of bacteria that is not a species of bacteria. That's a whole bunch of species of bacteria. That's like saying mammals. Well, that covers raccoons and skunks and deer and you and chimpanzees and mice, you know, but you're all mammals. So gram-negatives are a whole bunch of species of bacteria. Why did he say made gram-negatives so hard to kill? What do you remember, Alexa? Well, those are the genes. There we have the jumping genes. That's a whole different idea. We're going to pick back up on that. Hold that idea. What do you remember, Tori?
The armor that like surrounds.
They are wearing armor. It's like bulletproof vests on these little bacteria. And very often we see resistance because they have sort of armoring. They have some kind. So bacteria have a cell wall. We'll talk more about that in a little bit. This is frustrating. But if they have differences in their cell wall, sometimes it makes them harder to kill. Well, what determines how their cell wall is formed? Their DNA, their genes. Your genes, a bacteria's genes, a bluebird's genes, an earthworm's genes. I see your gum jay spit it out. A maple tree's genes. Any living thing's genes control how their body is built, how all their systems are built. And it is those underlying, those differences in their physiology that come from their genes, that come from their DNA, that make some of them resistant. Okay, so let's talk a little bit more about what bacteria are. Okay, so what are bacteria? What on earth are bacteria? They are single-celled. They are prokaryotes. A prokaryote, the cell has no nucleus, only a piece of DNA, some ribosomes, and a cell wall. They reproduce asexually, that's an A out in front of sexually, to produce offspring just like themselves, usually. There's an asterisk there. The reproductive process is called binary fission, and bi means two. Your bicycle has two tires. Okay, so prokaryotes, they are prokaryotes. And I had this conversation with a few of you yesterday. Pro is a prefix here. And we're not going to get into the Greek from which karyotes comes. But pro, think about a prototype. What's a prototype? It's an early model that you make of something. Like, I have this idea, I don't know if anybody here has anybody taken sewing with Mrs. Agnew? A few people? Okay. Anybody do any woodworking? Anybody ever build anything or make anything for themselves that you wanted to have in your house, wanted to have in your room? Okay. So if you're making something with, really expensive materials or fancy materials, you might want to do a practice run. One of my best friends made her own wedding dress, sewed it. She had never sewed a complete dress before in her life, and she decided she was going to do it. She was going to sew her wedding dress. And she bought really expensive silk, and she said, you know what, I'm sewing a cotton version first. Because she wasn't willing to take a risk of making a mistake on the really expensive fancy fabric, so she did a practice run. If you're building something in a beautiful, expensive hardwood, maybe you make it in a cheap pine first to make sure you kind of know what you're doing. A prototype is that first model you make. It's an early model. A prokaryote is an early kind of cell. These resemble probably the first cells that existed on Earth. They are very simple compared to fabulous complex creatures such as yourselves. My goodness, look at you. Aren't you something? You got multiple cells, they're working together, they're doing all kinds of things. That's pretty cool. These poor little guys, they got one cell. If it happens in that one cell. So, they don't have a nucleus. We're going to talk more about what a cell nucleus is, but it's the middle of the cell, it's the control center of the cell, it's where you store your DNA neatly. It's like keeping your money in a wallet rather than just throwing it randomly into your backpack. They have a chunk of DNA. That's it. It's a circular ribbon of DNA. They have some things called ribosomes. We're going to talk more about what those are later, but does anybody know what a ribosome is? Did you do some cell stuff in middle school? What is it? Do you remember? Okay, that's fine. You've heard the word. That's a good start. That's where you make proteins. You make proteins in a ribosome. And they have a cell wall, which if you did cell stuff in middle school, you probably talked about plant and animal cells. And animal cells don't have cell walls and plant cells do. But bacteria, prokaryotes, are not plants and they're not animals. They're their own thing. They have a cell wall. And some of them also have an extra special coating on the outside called a capsule. And those gram-negatives have a very strong protein capsule. They reproduce asexually. They just split in half. They copy all their DNA. They split the ribosomes. They split the cytoplasm. And that's it. They're done. That usually results in 2 new identical organisms. Anybody ever make a mistake in copying something? All the time. Actually, you're making mistakes right now. And you say, but I'm not copying anything right now. Au contraire, my friends. Your cells are copying DNA right this minute as you sit there. Dude! Okay, I'd like to invite everybody to take one real quick look at your hands. Wow. They're covered in skin, which is made up of tiny little cells, and one of those cells that you're looking at right now is dividing right this very instant. It's copying the DNA, and you know what? It's probably screwing it up. What? Are you saying I screw things up? Yes, yes I am. Fortunately, your body and the body of bacteria have, well, more your body, there are mechanisms in your cell that correct those mistakes, that catch them. Just like if you're copying notes off the board and you misspell a word and then you kind of mentally look back and you go, oops, whoops, there's not an E on the end of that, and you cross off the E. Your cells do that. Bacterial cells copy DNA and they make mistakes. And they probably catch some of them. I don't know, I honestly don't know how strong the mechanism for catching mistakes in bacteria is. I'd have to look that up. It's, outside of my experience. But they make mistakes, and when they make a mistake, guess what? The offspring isn't just like the parent bacteria. It's got a little bit of a difference. So that reproductive process, it's binary fission. It just means splitting in two. Fission means to split in two. That's all it is. Okay. The one question that I saw, it came up yesterday, so I added a special little thing to my notes. Is a bacteria the same as a virus? No. Viruses can't reproduce themselves. So here's a mean, nasty trick. You miss a day of school. You miss taking notes in, I don't know what class, and Spanish. And you walk in the next day and you look at your friend's notes and you go, dude, those are really sloppy. Those are messy. Your notes are not sloppy. You know that. You're way neater than I am. Those are horrible. You should recopy all of those. I don't know how you can even study from those. You should recopy all your notes right now. And your friend's like, oh, okay. And they recopy all their notes. And then you go, here, I'll take that copy. It's not so bad. What'd you do? You just tricked your friend into copying the notes for you. You know, you could have been an honest soul and said, hey, can I borrow your notes? I need to copy them. But no. Treachery and deceit. It's kind of what viruses do. They trick cells into copying their DNA. They can't even copy their own DNA. How stupid is that? So they're not considered living things. They're not considered living things because they can't copy their own DNA. There are a couple other important distinctions, but that's the big one. And antibiotics don't work on them. Antibiotics are against living things, against life, anti-bio. They don't kill viruses. So if you've had the flu, who's had the flu? Woo! I hate the flu. I get flu shots now. Before I got flu shots, I used to, without fail, get the flu every fall. I'd be down for 4 days, you know, miserable. The flu is a virus. You can't take an antibiotic and get rid of the flu. The common cold is actually a virus, though we often get bacterial sinus infections and we say we have a cold, but we really don't. The cold is a virus. Viruses are a different thing, and antibiotics don't work on them. And that's a really important thing to remember. We're going to talk about why that is later. Okay. Here's what we're going to do. I want you to get out your vocabulary packet. You may or may not have all of your A's and B's done. That's okay. They're not due until Tuesday. And if you've got them, I'll be happy to check them. But right now, we've talked about some words that are on that packet. And I want you to look at those words, and I want you to think about the discussion we've had. And I want you to see if you feel ready to fill in Part C on any of those. Do you feel like you understand what those things are well enough to describe it in your own words? And if you don't, that's okay. If you feel like you're not there yet and you need to understand more before you do Part C, that's fine. But if you feel like, oh, okay, I get it, I see what that is now, great, fill in Part C. So in that last 10 minutes, I'll have you do that. My goal had been, my thought process had been that we would have a recall quiz, but we're not quite where I thought we were going to be. So I'm going to shorten up that recall quiz because we didn't cover all the things that were on that quiz. And so we're going to do a quick little 2-question quiz, especially because we have a long weekend. I don't want to give this to you Tuesday morning because you won't even remember who I am or why you're here. So I'm going to give you a little 2-question boop boop for another assessment grade. So especially if you got a low one on the nightmare questions, this is your chance to redeem yourself. If you were here and awake and alive, you should do well on this, I think. So let me fix that. I'll post it on Classroom. And if you don't get it done before you walk out of this room, you do it at lunch.